let's get more towards the precision medicine part. And we're going to start with the golden child of precision medicine. Not you, Mark.
But it can be Mark, too.
It could be Mark, actually, but he's not a child anymore. Okay. Why don't you talk about advances in FLT3 mutated, especially for the previously untreated FLT3 mutated patient?
So yes, there's been a lot of activity in the field after a long slog through the wilderness. So we have the results of CALGB 10603, if I think I'm getting the names right, RATIFY, which was the randomized study of induction chemotherapy with or without the first generation FLT3 inhibitor midostaurin. And that, while it resulted in what many people thought was only a modest benefit in survival, it was real, it was believable, and in fact, as we've been alluding to with transplant, the patients who got that and then went to transplant, a big fraction of those seemed to be cured. So that was a big first step, that you could actually improve outcomes with this approach. Then along comes the next player, I guess, is actually technically quizartinib, because quizartinib is actually approved in Japan, and that's approved in the relapsed setting. But of course, fast on its heels was a drug, gilteritinib, and so you've got this approach initially directed at the relapsed refractory setting. But in the upfront setting, we've got midostaurin. These better drugs, or more targeted, more efficient drugs, second generation drugs, what you will, are being now studied in the upfront setting, head to head, typically with midostaurin, or in the case of quizartinib, against placebo. So there is a trial called Quantum First. Quantum First actually got started in the middle before we knew about the results of RATIFY, the midostaurin results. And it was actually carried out initially in the US before the RATIFY results were known. And then when the RATIFY results were known, where it's, ethically I have to give midostaurin to my patient, that's okay, because in fact, midostaurin didn't appear in the rest of the world for quite some time. And so the study finished accrual in the rest of the world. So we have a study out there, much like RATIFY, induction with either placebo or quizartinib, and you can actually get quizartinib after transplant. And quizartinib is a very different drug than midostaurin. It is, the opposite end of the spectrum. Doesn't work against the TKD mutation, but very potent, very selective, and might actually be the best drug to fold into the induction setting. So chaos is coming when the results of that are read out.
So let me come back to midostaurin in the RATIFY trial. It included both TKDs and ITDs, low and high allelic burden. Does it matter?
Not in the least. Well, yeah, actually, the FLT3 disease, TKD, high allelic burden, NPM1 mutated, which is a big modulator of how patients do, the disease behaved exactly as we expected it to in the context of the trial, in that if you had a TKD, you did better than if you had an ITD. If you had an ITD, you did better if you had less of it. If you had an ITD and you had an NPM1 mutation, you did better. But in all categories, midostaurin had kind of the same benefit marching on up. So it really didn't matter. Midostaurin should be used in all categories. Prognostically, it's interesting to know, but it doesn't influence your initial outcome, or your initial treatment.
Rami, I'm going to put you on the spot about something. So while the FLT3 inhibitors are being developed like that, there's also been continuing work just trying to figure out how to prescribe 7 and 3 from 1973. And so there have been large randomized trials, and to summarize 3 of them, the ECOG 1900 trial, to the MRC trial with 90 versus 60 of daunorubicin, the ECOG 1900 was 45 versus 90, and a Korean trial of 90 of daunorubicin versus idarubicin. Those studies showed that in the FLT3 mutated subset that there seemed to be a benefit for 90, but yet midostaurin was developed on a backbone of 60. Does it matter? And what do? Do you use a higher dose of daunorubicin?
No, so yeah, I totally agree. So like those studies were done in the context where we really did not have novel therapies or active therapies. So the question was always 45, 60, 90. And clearly with the 90, at least with the ECOG intergroup study, that there was a advantage of the 90 in the FLT3 mutant. with the midostaurin study, with the survival benefits shown in that patient population, the study was designed targeting that population, we've kind of adopted what's done on the study going down to the 60, because obviously, as you mentioned also later on, the studies suggested that 60 is probably as good as 90. The original ECOG study was 45 versus 90 daunorubicin. So I think in this era, like, we are probably beyond looking at the anthracycline dose. Actually, hopefully, in many subset of patients, we are going to go away from the anthracyclines.
I tell my fellows, if they're in a room with people talking about what dose of the chemotherapy you should use, you should leave. You should just leave.
Just go to IDA. Just do IDA. Leave it.
That's what we do.
That's what we do.
That's why no one ate dinner with me last night.
But like fellows and stuff sometimes ask us, like, why, what dose, what, I think we've just adopted, if we are doing anthracycline, the daunorubicin 60. I still, in elderly patients, I think idarubicin is good equivalent choice. I don't think we've ever showed that daunorubicin better, but we could talk about this, but I think we have more exciting stuff. So